Today we have the pleasure to introduce you Dr. Rex Bernardo.
Thank you so much for your hospitality in having me here today. As mentioned, my name is Rex. I'm a professor at the University of Minnesota in the United States. Just a little bit about myself. So I've been at Minnesota for 25 years now. Before that, I was at Purdue University in Indiana. Before that, I worked for 9 years at a private company. My wife and I have 6 F1 progeny, okay, and 3 F2 progeny. I call them F2. Those are my grandchildren. So I'd like to acknowledge especially 2 really good friends who I value and respect. One is Dr. Eduardo Brun, who is here. Eduardo and I were in graduate school together at the University of Illinois. And he was a really smart guy, this guy, Eduardo. When we had problems with statistics or things we couldn't understand, we go to Eduardo because he would patiently explain, like a very good professor, for the things that are going on. And I'd like to acknowledge Dr. Patricio Mayor. Patricio is a graduate here, and he was my former PhD student. Why I'm here? I'm director of our plant breeding center, and we used to have get a lot of good students from South America. Many apply to our graduate program, and now the numbers of applicants we have from South America have decreased. And we'd like to build that up again, because the students who we've had from this part of the world, they've always been really excellent. So I'm just letting you know why I'm here. It's a bit of a selfish interest, actually, but good for us and good for you as well, professionally. So I'm here to talk today about what I call a plant breeding triangle. And by the way, I'll have slides, but I noticed that when I have the traditional slides, you look at my slides, but you don't look at me. And I'd like you to look at me and maybe pay attention to me. So this is a different kind of talk, different way of giving a seminar. I'm just speaking to you just like this. Plant breeding triangle. Who here are plant pathologists? Any plant pathologists? Okay, good. So you need, correct me if I am mistaken, but in plant pathology, you have a plant disease triangle, right? And the pathologists are really creative and smart. They have this disease triangle to summarize key concepts in plant pathology. Meaning to say that you get a plant disease, you need 3 conditions to be present. Number 1, you need a susceptible host plant. Number 2, you need a pathogen that is available to infect, a virulent pathogen. And number 3, you need an environment that is favorable for the development of the disease. You need all 3. If you take one out, if the environment is not good, if you don't have a susceptible host, if you don't have the pathogen, you won't get the disease. And so I thought, you know what, in plant breeding, we can think of what we call a variety triangle. And also in plant breeding, there are 3 ingredients before you can develop a good variety. And here this morning, I'll talk a little bit in general about these 3 ingredients that we need. And these are not new, but it's mainly to get us thinking and increase our appreciation and perhaps think about what the future would be. So the 3 components then of this plant variety triangle are, number 1, you need good germplasm. You need good germplasm. That's the ingredient, that's the secret sauce to success. You need good germplasm. Number 2, you need good breeders, okay? And number 3, you need tools, resources to run your breeding program. Take one out, you can't get a good variety. You can have the best breeders. You can have the most advanced resources. Without good germplasm, you won't be successful. Obviously, without breeders, you won't be successful. You might say, oh, but now we have artificial intelligence. We have AI. Maybe machines can do the breeding. I think machines can do part of the breeding, not everything. And of course, without tools and resources, you can't be successful either. So I'd like to, again, just talk about these and maybe initiate a discussion of perhaps what our views in the field are and where things are going. I am interested, I've always been interested in technologies and in methodologies. Even when I was a young graduate, I got my degree, bachelor's degree in the Philippines, and I was hired as a teacher for laboratory courses, and I started working in a national breeding program for sweet potato. And when I was hired to be part of the sweet potato breeding program, I was interested in how can we make this program better. And of course, I didn't know too much. I had a bachelor's degree and I wanted to study more. But I noticed that my advisor who was leading the breeding program, a key trait was specific moisture content of the sweet potato. You don't want it to be too wet, okay? And the way he was evaluating the moisture content of the sweet potato, he had a knife in his back pocket. He would take out the knife and peel the sweet potato because it was dirty, and then he would bite it. And go, that's too wet. Don't like that. He would take the next one, bite. Okay, this is a good one. And I go, there has to be a better way, maybe, of evaluating sweet potato in the field for the moisture content. So I found, for example, that the moisture content is correlated with a specific gravity. And so what I did was, I said, what if we develop a system, we have a solution of salt, and then in this salt solution, we put the right amount of salt so that our check, our standard commercial variety, it would sink, okay? And then that becomes a screen for moisture content. Because if it has a high moisture, then that sweet potato will float. So we did that in the field and we started using that. And they go, oh, okay, that works, because now it was actually nice to see some sweet potatoes sinking, and those were selected, and some sweet potatoes that were floating, and those were discarded. And I saw my boss, he was using that breeding method, and he was very happy with it. He didn't need to use his knife anymore and bite. And I said, okay, that was a success. That was an example of a very simple, low technology tool that was applicable in a breeding program. I did not publish it. I don't think it was publishable. But it's too simple. People say, it's too simple. We don't need to publish that. And I don't think you need to. Now, when I became a graduate student at the University of Illinois in the US with Eduardo, breeding was very easy. Easy. It was, selection was all phenotypic. All you had to do, you grow your corn, your maize in the field, you evaluated it for yield, for grain moisture, for the height, and then as a breeder, you said, okay, this is a good parent, this is a good parent, I cross good by good. If I cross two good parents, chances are, even if I'm not a very good breeder, if I cross good by good, I'll get something good in the end, because the parents were good. And then you repeat that process each year. Cross good by good, try to find something better, repeat that process. And our lives are changed. Eduardo was there. I still remember in that in the basement of our, there was this guy, a colleague, a new PhD student from Spain. And we were in quantitative genetics, and this new student was in molecular biology, molecular genetics. And he told us about this new tool called RFLPs, restriction, fragment, length, polymorphisms. Does anybody even know here or remember what are? Okay, I get it. So 2 people. Okay, 3. Maybe the older people know about RFLPs. And for the younger people here, go, what is that? What's RFLPs? You know, now we have sequences. We have DNA sequences. Well, RFLPs. Well, RFLPs, they look like this. Yes, you had to run gels. You had to do electrophoresis, run gels. You take a X-ray, you know, autoradiogram, and then those are the bands you got, and then you needed to score them manually. Okay, you look at this line and you go, oh, is there a band there? Is there not a band there? Are they actually in line with each other? So that's how we did things, right? You had to score it manually. And so this is the product, but there's a lot of work that went into getting this type of data. For example, I still remember, I think you were there too, Eduardo. We were called by our advisor to the field, and they were told, advisor told us, today we are going to collect leaf samples for DNA analysis. So we reported to the field. We were given gloves. They told us, wear gloves because we don't want your DNA to contaminate the corn DNA. So we wore gloves. We were given scissors. Today, I think you get, leaf punches for little leaf samples. No, we were given scissors, and we were told to cut whole leaves about that big, 3 or 4 leaves that big of the plant. And then we were told to put it in a bag, put it in a cooler. The cooler was taken to the local airport where there was a private airplane that was hired to take the leaves to the lab in the next state of Iowa. I go, wow, this is expensive. How can this work in practice? This is too expensive if you need an airplane to take the leaf samples. That you're afraid, just driving it by car for 6 hours, that's too long. You really need an airplane. And our advisor very wisely told us, Rex, Eduardo, I want you to think of the day when we won't need to do all of this logistics for this type of work. And at that time, the cost of one of those bands, so to speak, was maybe about $5, $10. He told us, think of the day when the cost of one of these data points is less than the cost of a paper clip that we use every day to pollinate corn. And I said, no way. No way is that going to happen. That the cost will be less than the cost of a paperclip. But guess what? I looked at the cost of a paperclip last week. It's now less than the cost of today of getting a DNA fingerprint, single nucleotide polymorphism fingerprint. What's the lesson here? The lesson is that when we think of the resources that we have in a breeding program, the technologies change, the cost changes, and so we should not limit our thinking to what we have now, but we should have an open mind and think of how might things change in the future, because that if we're limited to what we are now, then we cannot explore the possibilities that might be there in the future that would be made available by advancements in the technologies we have. And so I know that not all of you are in plant breeding, but that is a lesson. Whatever field you are in right now, try to think of, well, this is what I'm used to, this is how things are, these are the costs right now, but how might things change in the future? They might change, they may not change, and that's okay. But we need to think about possible changes in the back of our minds. Because it's not only with marker technology that has changed. Another big technology that has changed in the 40-some years of my career is computation. I am in the field of quantitative genetics, statistical genetics, computers. It used to be that the computers we had back then were these big machines, mainframe computers. And now, of course, we have our even laptop computers, and we have these things here, smartphones that can do incredible things. So how does this change then the way that we can do breeding? We can think of, well, now I have my smartphone. What are apps? What are tools that could be developed in my own smartphone to help me with my work? If I'm a breeder, for example, can I use a phone and camera and scan the plant and tell me things about the plants there out in the field? And can I use artificial intelligence to tell me things that I need to know? Talking about computing, when I started my career in a industry breeding program, this was the time when we had Microsoft DOS. Anybody remember Microsoft DOS? Okay, the same people who remember RFLPs remember Microsoft DOS. DOS was the time when you turned on your computer, you just turned on the switch. It was on. When you were done with your work, you switched off the computer, the switch, it was off. That's how it was. And when Windows came, I remember when Windows came, my first thought was, why would I need 2 or more applications open? Why? Why do I need to have Excel and Word open at the same time? I just work with one at a time, one application at a time. And now it's amazing that I was thinking that way when today I have, several applications open at a time. But at that time, the computers had only, about 8 megabytes of RAM. 8 megabytes of RAM. And I remember that for some computations I was doing, I needed to have a lot more memory. And this was before you could order things online. So I actually had to order a computer by picking up a phone, looking at the catalog and picking up a phone. And I said, I'd like to buy a computer with 64 megabytes of RAM. Okay? And there was silence on the other side of the line. And she asked, what do you need 64 megabytes of RAM for? So I had to explain, I'm doing these scientific calculations. I need a lot of memory. I really need 64 meg of RAM. Please don't question my choice. I know what I'm doing. Now you order a computer, 64 gig of RAM. That's fairly common. So meg, gig, that's 1,000-fold increase in memory. Which reminds me, going back to the data points, this study for which we were collecting leaf samples, at that time, that was state of the art. The number of markers, 34. 34. 34 markers for 10 chromosomes. That's only 3 or 4 markers per chromosome. Today, in some studies we do, we have 34,000 or 30,000 markers. That's pretty common. So you have 1,000-fold increase in the computing capacity, 1,000-fold increase in the number of markers. We can do a lot more things today. And this leads us then to questions, looking at the future at, what resources might we have? What are some technologies that you think you hear about these days? Let me open that. What we talk about, there's this new technology here. What comes to mind? What do you, CRISPR. Oh, yes. Funny thing is, things come in what I call as bandwagons. So back in, the 1930s, some bandwagons were mutation breeding, where we can bombard our plants with X-rays or treat them with chemicals to have mutations, and we could have really big, big grapes or big plant parts, and that was one bandwagon that went up, and then people started doing it and realized, it did not meet the promise, and so it came down like this. But the funny thing is, in science, when one bandwagon comes up and then we begin to get discouraged, the next bandwagon would come up. There's a new thing that happens that occupies our attention and that draws resources. So CRISPR is one way of mutation breeding, that we're back to mutation breeding, except now it's not random mutation. It's for gene editing. It's very targeted. So thank you, CRISPR. I had the chance, just side story, I had the chance to consume some gene-edited products actually in my lifetime. This was several years ago. I was in a lab in Sweden, in the lab where one of the inventors of CRISPR, she worked. And she said, here's some gene-edited cabbage. Would you like to taste some gene-edited cabbage? And she said, sure, I'll taste some gene-edited cabbage. So I ate it. It did not taste any different. So I think I'm still fairly normal. So I think it's okay. All right, other technologies that you hear about today. Yes, okay, so technology, we have sequencing technologies that are available. It's interesting how some technologies might make other technologies obsolete or no longer there. For example, in the late 1990s, RNA microarrays were a big thing, okay? And there was a lot of studies, a lot of literature even on how to conduct experimental designs with microarrays. Guess what? Sequencing came. Nobody talks about the RNA, the microarrays anymore because of sequencing. Okay. Others. How about drones? You know, drones are a big thing right now, right? So you have drones that take measurements of plants, and what we can get, the measurements of plants, they vary. And I asked breeders, some of my former students who work with companies, how do you use drones in your breeding program? And they told me, well, one common use is to count the number of plants in a row. And I go, really? That's it? Yeah. We use it to count the number of plants in a row. I mean, we need to know how many plants are in a row, right? You have 20 plants or 25 plants. That affects what we do. But I said, oh, it should be more exciting than number of plants in a row. Well, yeah, but that's really a really important use. Well, talking about more exciting uses of drones, I also work with an apple breeder at the University of Minnesota. And in apples, what's most important is quality. Quality is the number one objective. Yield, secondary. They don't even evaluate yield. I asked our apple breeder, how do you evaluate the yield of apple? He goes, oh, it's really easy. You go, you look at the apple, you ask, does it have enough apples? Yes? Oh, okay. Next tree, does it have enough apples? Ah, no. Then you say no. That's how you evaluate. It's yes or no. But we figured, well, you could do maybe something better. Hello. With drones, I mean, yeah, with robots, they're developing robots now that would go around the orchard, and it would take images of the apples, and it would separate the red fruit from the green fruit. And based on the size of those, red things, they would estimate the yield of the apples. And I go, okay, to me, that's a more exciting use of drones than just counting the number of plants in the field. But now I need to ask my apple breeding colleague, what if it's a green apple? It works for red. What if it's a green apple? How will it work? I should ask him that. For me, give me a drone that's underground. Okay? I like an underground drone. I don't think that exists, right? But I'm interested in studying the parts of the plant that I cannot normally see. And how it's typically done is called shovelomics. You get a shovel, you dig the plant, and you see how it is. But I figured, you know what? If there can be little robots, little soldiers that would go under the ground and penetrate and report back to the mothership how those plants, how the roots look like, I think that would be a wonderful application. I think the way to do that is you tell an engineer it cannot be done. Yes, tell an engineer it cannot be done. And if you talk to the right engineer, the mind would be, oh no, I'm an engineer. I can invent anything. And so maybe that's the way to do it. Today, we're thinking also of I mentioned artificial intelligence, I think is a tool that's out there. And the question is, how can we use artificial intelligence in a breeding program? Can it replace breeders, actually? Or maybe let's not go there yet. Can it replicate, mimic some of the breeders' decisions? And I think the answer is yes. In fact, one of my PhD students, part of his thesis is trying to look into machine learning and trying to see how well machine learning can replicate breeders' decisions. So the experiment we have is, well, we have these data sets, and we're going to ask several breeders, 5, 6, 7, 8, 9, 10 breeders, to each select which corn lines do I keep based on my own criteria, okay? And then we're gonna ask a machine to say, okay, I want you to learn, and how well can you replicate the breeder's decisions? I think it can be done. It'll be interesting to see, okay, does it work better for some breeders than for others? Are some breeders more replaceable, so to speak, by a machine than others? Not that we want that to be our criterion, because breeders will know, oh, I don't want to be replaced by a machine. I'm going to select at random over here. No, we don't want to do that, right? So anyway, but I think, again, there's a place for AI. And when we hear of AI these days, oftentimes we're scared. And even as a professor, sometimes I struggle with AI because I teach a course for first-year students, and it involves some writing. And I enjoy reading the writing of a first-year student because it's not perfect. Like, oh, that's cute, and there's room for improvement. Sometimes I come with a look at the paper, I go, this is too perfect. I said, no way a freshman student could have written this paper. But then for me, it's more, well, I need to, the burden of proof is on me. And it raises also questions even for our master's and PhD students. One of my PhD students approached me and said, Rex, what's your view on ChatGPT? Is it, how can I use ChatGPT as I write my thesis? And if so, how? Hey, let's take a poll. Who thinks it's okay to use ChatGPT to help you write your thesis? Can I get a raise of hands? No pressure. I get 1, 2, okay, 3, 4. Who says no way you should not use ChatGPT? Okay, we have some as well. Here's what I told my student. I want the ideas to be yours. The ideas absolutely should be yours, no question. Ideas are yours. If you use ChatGPT just to improve the quality of your writing, the English, the flow, the transitions, I'm fine with that. Because I said, it's like you send your paper to a copy editor just to improve, the quality of the writing. That is my personal view. Not everybody in my university subscribes to the same view. But anyway, it's worth discussing. It's worth discussing, I think, in any university setting. And it'll be interesting to see, I'm just mentioning the tools and resources there right now, what the future holds. You have AI, well, what comes after AI? Maybe we'll be back to natural intelligence. Well, no, it'll never disappear. Okay, so I've talked about tools and resources. That's the first ingredient. A second ingredient that we need is germplasm. Germplasm, it's not something I've paid too much attention to. Oh, I actually skipped a slide here. We used to store data in these things, right? Floppy disks with 640K of RAM. It says 1986 data. That was my PhD thesis. I don't know why I'm storing I don't know why I still have them. I can't read them anyway, right? I don't have the computers to read them anymore, but I spent a lot of work getting these data. At least I have some copies somewhere. Okay. I'm sorry, one last thing. There was mention about CRISPR, about tools and technologies. This is a slide I gave at the University of Wisconsin in September 2001. And I posed the question, I had a car engineer friend, how do car engineers design cars, and how does that compare to the way a breeder creates a new variety? And I said, well, if I'm a car engineer, I would try to understand how each car part acts, how each part interacts with the other car parts, the engine with the fuel injection, with the gears and so forth, and how related parts interact with the environment, including the driver. And I would use all of this knowledge to create a design, to create a better car. Now, however, if a breeder, I thought, were to design a car the way he or she breeds plants, this is the procedure. You take two high-performance cars, a Porsche and a Ferrari. Then you shuffle those car parts and create thousands and thousands of combinations. And then you select the best shuffled car and you repeat that process. And this shows us just the difference between a design process and more of the process we use as breeding. We have some control. We can control which cars we cross. You won't cross a Porsche with a really bad car, okay? We can control the manufacturing process, the process of shuffling, the quality control through the field trials we use. But I'm saying here that we create thousands of these combinations because we have no control over meiosis and fertilization. Today, that might not be exactly the case. Actually, today, there could be ways to control recombination. It's an active area of study. For example, the genes that control recombination are known, and so we can either increase the rate of shuffling or decrease the rate of shuffling. If you already have a good combination of parts, you don't want to shuffle those anymore, so you could decrease the rate of shuffling. CRISPR, we typically think of CRISPR for gene editing. CRISPR can be used to control recombination, actually induce recombination. You could use CRISPR, it creates a break in the DNA precisely where you might want that break to occur. So now it's possible for a breeder to ask the question, well, where should recombination occur in the genome? And can we induce a recombination at that point to get exactly the product that we need? So here, about 24 years later, I say this slide, it's still largely true, but maybe in the future, it might not exactly be true anymore. And again, why? It's because of the development of tools and resources that help us in the work that we do. Sorry, I forgot those slides. But next topic of germplasm. Corn, sorry for the corn breeders here. Corn is kind of boring. Yeah, yield, moisture, height, disease resistance. But you can't really taste it to enjoy it, or at least the different varieties, right? For me, sweet potato was a bit more exciting to work with because I could taste the sweet potato and look at the differences in the colors of the leaves and vines and so forth. This was maybe my first work involving germplasm was dwarf corn. I said, well, if you look at all the cereals, they've become short over the years. And what if we make the corn purposefully short by introducing dwarfing genes? And then we can, the plants will be shorter, the ears will be smaller, but we can grow it like wheat, like a small grain. Put in a lot of plants and then harvest it and maybe increase yields by a lot that way. I did this research in the 2000s. After a while, I did not pursue it, and I forgot about it until, guess what? Bayer has introduced short corn. And I go, oh, I didn't have the resources to do so. But I said, I wish I had worked on that a bit more. But that was my first work, so to speak, with regard to that part of the triangle that has to do with germplasm. Now, I'm in the stage of my career now, though, where I'm looking at other things, and I'm beginning to be more interested in germplasm. Part of that is when you look at that breeding triangle, and if you look at the major players, the major breeding programs, they all have good people. They all use pretty much the same kinds of approaches, methodologies, differences in details, difference in scope. What would differentiate them is the germplasm that they have. It's either you have the germplasm or you don't. And so that is one, actually, that part of the triangle that I think really, really needs a lot more attention. How can we increase the germplasm that we work with? Now, lately, in terms of germplasm, I have been working with on different species, actually. I started now working kind of like a vegetable breeder. I'm now doing some work on vegetables, which to my mother, before she passed away, she knew I hated vegetables as a child. And so if I would have told her, hey, Mom, I'm now a vegetable breeder, she would go, wow, there is hope in the world because you can change, right? But now I'm interested in vegetable breeding. Part of this has to do with my work as director for Plant Breeding Center. A few years ago, I read an article in the local media about farmers markets in Minneapolis, St. Paul. It's a big city A farmer's market is where local producers would bring their fruits and vegetables within the city, and then people from within the city would buy those fruits and vegetables. I read that some of the local farmers began producing leafy African vegetables. And some people would drive, say, from Texas or from Seattle, Washington. That's about a 12-hour drive, one way. They would drive for 12 hours to buy these leafy vegetables, then drive back. Like, wow, you would drive 12 hours just to get leafy vegetables? I can relate a little bit because the foods, if you're an immigrant like I am, the foods you grew up with, they remind you of home and they're meaningful to you. When I go back to the Philippines, I tell my friends, do not take me to a fancy Italian restaurant. Please don't. Give me the street foods that I grew up with. And it's the same with our immigrant communities. And leafy vegetables are, of course, best consumed fresh. And so we started this breeding program for leafy African vegetables that is led by our students. It's a fully volunteer breeding program, student-led, student-driven. And so our students identified vegetables that they can work on. They've started building relationships with immigrant communities. They have a community garden where they separated it into two groups. They have 4 species that they focus on in research, and they have a community garden of these different rich species that people are interested in. So I saw cassava growing in St. Paul, Minneapolis, St. Paul. I never would have thought I'd see a tropical plant growing there. Somebody even tried to grow a pineapple. Well, I learned that, yes, he used the greenhouse during the winter to grow the pineapple, then transplanted it in the summer, and that's okay. But as you can see, it's been a very meaningful breeding project. And now, because we're looking into these vegetables, acquiring germplasm actually becomes key to the success of this breeding program, because, well, these are African vegetables, and how well can they grow in Minnesota? And so that's just a photo from the field day. What our students do is they also have these samples of the vegetables that they give to people who come to the field day. We also share a meal together. We ask the local immigrant communities to bring foods from their cultures, and we have a wonderful time doing that. And again, it just expands our horizons in terms of how we see plant improvement, and it brings us a bit closer to the people for whom these plants are important. And so this topic then brings me to people. You cannot have a successful breeding program without good people running it. And when I look at plant breeding today, and I think for any field for that matter, I can make that statement, there's a lot to learn, and I can say that in a positive way, and I can say that in a negative way. I can say that there's a lot to learn. This is really exciting. You can learn about this and that. There's a lot to learn. We're in an exciting time. But I could also say that in a negative way. There's a lot to learn. Do you feel that as students? There's just too much. There's too many papers that are published these days. There really are too many papers that are published these days. I don't know about you, but I cannot keep up. There's a lot to learn. And I used to rely on my students to tell me what papers I should be reading, but they also, it's like, there's just too much. Too many review papers, too many papers published here and there. And I actually asked, I was at a meeting one time of an American Seed Trade Association. I asked them, what are things we ought to be teaching students, new things we ought to be teaching students today? The answers came quickly. Students need to learn about CRISPR. Students need to learn about drones, phenomics. Students need to learn about artificial intelligence, machine learning, at least a little bit. I said, okay, what do we take out? Because in our curriculum, students take a fixed number of classes. a fixed number of credits or units. If you want new courses that students need to learn, we need to take something out to accommodate these new courses. Guess how many answers I got? 0. Meaning to say, well, yes, we keep what we have, we keep new things. That is not sustainable. So, and I think for any given field, including plant breeding, we need to seriously think, well, what are things that really, really, really are important, and we teach that in our breeding program. And for the rest, well, we give a good foundation and allow students to learn those things on their own without necessarily teaching a course. But I need to mention, though, one specific thing that I think students today really, students and professionals really need to learn. And this came about, I sent my former students an email saying, what are things that you wish we taught to you at the University of Minnesota, but we did not? And the answers came, and they all had to do with soft skills. They all had to do with professional skills. And because of that, I started developing a course called Professional Skills of Scientists for Scientists. I'm going to Uruguay after my trip here. I'm going to teach a short version of that, first time at the University of the Republic. But in this course, we talk about differences among people, how different people have different strengths, for example. We talk about conflict and managing conflict. We talk about how to manage yourself, how to manage your boss, how to write a good email, how to run a meeting, work-life balance. These, again, soft skills. And I think that as students, as scientists, these are things you need to pay attention to. And I tell my own students, you know what? When you graduate, nobody is going to look at your transcript of records. Nobody's going to say, oh, you got the A here, oh, you got the B here. No, nobody's going to look at your grades. I say, you graduated from the University of Minnesota. Your employer would know that you are technically good, competent, you're very good. What they would focus on is how well can you function as part of a team. And so your interview questions would be, well, can you tell me about a time when you struggled with your boss or you had a conflict with somebody else? Those are the kinds of behavioral questions that you would encounter in the workplace. So that's, again, that's my encouragement to you, to keep you thinking, if you're faculty or students, okay, what can you do in this area of developing soft skills? Okay, one last thing I need to talk about. Breeding triangle, resources, germplasm, we need people. One more thing we need to talk about is that importance of that triangle itself. I think as a plant breeder, I need to continually talk of how important that triangle is. In particular, how important the middle of that triangle is, which is the improved variety. We live in a society in which appreciation for food and agriculture is not as high as it was in the past. And I know I'm talking to a different audience here. You are all in this beautiful campus devoted to agronomy. And so you know where the food comes from. But oftentimes, say in the United States, you might ask a small kid, where does your food come from? Oh, it comes from the grocery store. It comes from McDonald's. And for them, that is the extent of the supply chain that somehow magically the food arrived at McDonald's or at the grocery store. And so I think we, as agriculturists, as plant breeders, need to continually communicate the importance of what we do. And we need to be creative. We need to find creative ways to communicate that. Now, I think I found one way to do that, and I'll just give that as an example. So this is the flag of the University of Minnesota, and these are the colors of the University of Minnesota. Maroon and gold. If you live in the state and you see maroon and gold, that's branding. Immediately you identify, okay, that's the University of Minnesota. Are there school colors here in the university? There are. What are the colors you have? Green. And just green. Green and white. Okay. I think something can be done. Okay. No. When I say something can be done, so we have these as the school colors. I developed this variety of sweet corn. Maroon, gold. So I show that. University of Minnesota. Sweet corn. And what I do is I use this as a vehicle to communicate plant breeding and the importance of plant varieties. Economically, this has really no value. It's a curiosity. I do get pressure to release it as a variety, and I'm hesitating. I say, number one, it grows well in our farm where we have good soil, we have good fertilizer, we have good irrigation. If I release it as a variety and it's grown somewhere in the state and it doesn't do well, they'll say, oh, Rex Bernardo, he's a really bad breeder. I mean, maybe I really am a bad breeder. And it's all cultural, right? So that's one reason. Second, it's higher value, I think, as an educational tool. Because what I do is I use this as a vehicle to tell people about plant breeding. I invite people to come to my field and take that corn and taste it. It really is, you can, if it's really good sweet corn, you can think of it as a salad. It really is sweet and tasty. So I invite, couples, I invited kids, and we have fun, and I tell them about plant breeding. I say, okay, we have apple varieties, apple varieties, this is a variety, but we do a lot more work than apples or corn. We do wheat breeding. We breed for soybean. We breed for potatoes and other crops. So if I say, hey, we developed a new variety of wheat, nobody cares, at least in the city. But then I can use this as an avenue to tell people about the importance of plant breeding. I tell them, well, plant breeding is like a game of Survivor that we have on American television. And in Survivor, you have a bunch of people, crazy people, I think, who are, say, left in a desert island and they need to survive. They need to do challenges. And then somebody gets voted out. They need to do another challenge until in the end, there's one winner. I say plant breeding is just like that, except instead of having 20 or so contestants, you could have 2,000 contestants. And then they go through a challenge, a first year of trials. And then that 1,000 becomes a few hundred becomes a few dozen, you have one winner in the end. And then I show them this is the parent of this, the two parents of this F1 variety. You cross them, wow, you get a nice hybrid, and it looks like this. And so one day I emailed our university president, and I told our university, the office, no, I cannot email our president directly. I can email her office. So I emailed, I said, hey, would President Gable be interested? Can I bring some sweet corn to our university president? And I got the reply back, well, our president is driving around campus today. Can she come to your building and get the sweet corn herself? I go, wow, I get an audience with my university president. And so I gave her this box, and I got to talk to her about the work that we do and why it's important. And later on that evening, I heard that she took this to another meeting. She was very happy, and she talked about this plant breeding work we're doing. And then the Board of Regents also got to sample this. So the point is, in food and agriculture, in plant breeding, we just can't let the varieties speak for themselves and say it's good. We actually need to tell people about the importance of what we do. And again, why continued investment in this plant breeding, this cultivar triangle, why that would continue to be important now and in the future. Well, thank you so much. You have been a lovely audience. Thank you so much for having me this morning. I appreciate the time here.